Hello, this is now our lecture number 17. We're going to be talking about the action of drugs at the synapse and learn a little bit more about what happens biologically when you are exposed to certain types of drugs. And again, this is all along the lines of trying to understand the biochemistry of behavior. So you really have to understand what's happening at the synapse when you talk about drugs. So that will be our objective in this lecture. So this really is an emerging area in many ways. It's one that's constantly changing. It's one in which there's lots of new information all the time. So I want to try to give you kind of an overview of where things are at right now. There's an awful lot of work that's being done on investigating the biochemistry, for example, of personality. A lot of work that's being done on in the area of drug research about the brain being hijacked, for example, by drugs and what's happening in terms of addiction. Uh, Clearly, this is an area of great importance in the field of behavioral neuroscience. And again, my goal is to try to give you, a little bit of a glimpse of what is going on in that area. So drugs in the synapse, when we study the influence of different types of drugs, we really want to know what's happening at the level of the synapse. Again, neurotransmitters are being altered in some important ways by drugs. In some cases, those drugs are actually mimicking neurotransmitters. They might be changing the way in which receptors are responding to those neurotransmitters. And these are things that can happen at many different points of the cell, of the neuron, not just at the synapse. So these are, important issues, and I think we want to, understand some basic terms here. And that, first of all, to understand what we mean by drug agonists and drug antagonists. I try to simplify this. Drugs can either promote or they can suppress activity at the synapse. They can facilitate or they can inhibit that activity. Antagonistic drugs block the effects of neurotransmitters. And again, here, an antagonistic drug, and I'll be giving you lots of examples of this as we go along. An antagonistic drug is one in which, for example, this drug occupies this receptor site and it's completely blocking the receptor site from showing either excitation or inhibition. Agonist drugs, on the other hand, increase the effects of neurotransmitters. So here we have an agonist drug, which is actually just fully activating that receptor. And certainly we could have a competition that's occurring here between the two, of an agonist and an antagonist, in which the activation that you see might be less than what you would ordinarily see with the full activation by an agonist. So again, antagonistic drugs are blocking neurotransmitters. Agonist drugs are increasing the effects of neurotransmitters. Here's a really interesting example of some research that has been done in the area of learning and memory. This research being done by Joel Martinez, and Martinez has been using a apparatus that's called a Y maze that you see right here. And essentially what Martinez has done is he's trained this animal, in this case it's a mouse, that's been placed in the starred area here, and he's going to bait either this side that we see here or this side that we see here, and is going to train the animal to go to the side where the bait is. So for example, an animal in which we consistently place the bait on this side learns very quickly that when it gets to this choice point, that it's going to go left and is going to go to this side of the Y maze in order to get the food. Alternatively, we could take an animal and train it to go to this side because we place a bait on this side. So animals learn this really quickly. And one of the things that we know from Martinez's work is that acetylcholine is very important for remembering this response. So if we trained an animal to go to either the left side to get the food here or to the right side to get the food here, if we train those animals and then we started to inject drugs to them that either promote or suppress acetylcholine, we can observe the effects upon the ability of this animal to remember what it had learned. And indeed, what he finds is that when he injects scopolamine to these animals after they've learned the response, scopolamine is an antagonistic drug. It's decreasing acetylcholine. What he finds is that the animal, now when tested, let's say the next day, doesn't remember the correct side to go to. It has to be taught all over again. What Martinez has also done, which is so interesting, is that he takes another drug called physostigmine, and that's an agonist drug, and it actually increases acetylcholine production in the brain. If he takes an animal and trains it in this Y maze, so that it's performing really well, and then injects it with the physostigmine, and then let's say the next day tests the animal, what he finds is that this animal actually shows improvement in terms of memory. So again, he's able to selectively manipulate memory by manipulating acetylcholine by way of either a drug agonist or a drug antagonist. Very elegant work, very important work that's being done in this area. So examples of agonist and antagonistic drugs: L-DOPA is a drug agonist. It promotes the synthesis of dopamine. We'll talk about L-DOPA when we begin to talk about Parkinson's disorder. Reserpine is a drug that inhibits the storage of norepinephrine, and it does so in those synaptic vesicles that we've talked about. PCPA, a drug that's been around for a long time, is a drug antagonist, and it's inhibiting the synthesis of serotonin in the brain. Cocaine. What does cocaine do? Well, it blocks dopamine reuptake. Hence, it makes a lot more dopamine available in the synapse. So it's a drug agonist. So again, we can classify drugs as being either agonists or antagonists. Another important concept here that you have to have a little bit of background in, very important, is how drugs are classified in terms of what we call affinity and efficacy. Now, when we talk about affinity, what we're talking about is the ability of that drug to act on a particular receptor. That is, does it actually bind to that receptor really well? In other words, does it closely bind with that receptor? And that can vary. That affinity can vary from either being very strong or weak. The efficacy of a drug, on the other hand, is its tendency to activate the receptor. So a drug can have high affinity. That is, it can bind really well to a particular receptor, but it can have very poor efficacy. That is, it doesn't really activate the receptor. So one of the first things that people that are involved in pharmacokinetics, psychopharmacology do is they classify a drug in terms of its affinity and its efficacy. So those are two very important defining features of any kind of a drug. So drugs can operate on many different levels. Again, let's revisit this very important figure that we saw in our previous lecture. Drugs can increase or decrease synthesis of a particular neurotransmitter. They can cause those little packages, those vesicles, right, to leak, to burst open prematurely. They can either increase or decrease release that's occurring in this postsynaptic, in this presynaptic area. It can increase or decrease reuptake that occurs, right? They can block the breakdown into inactive chemicals. can directly stimulate or block postsynaptic receptors. So here's 6 different ways in which drugs can work. And one of the biggest challenges in the drug industry is to develop drugs that are only going to have one action. That is, their side effects are going to be very limited. They're only going to be operating on one part of the cell. And that's what a lot of drug research does now. And this becomes particularly important when we're using those drugs in order to treat behavior disorders, for example. So let's take a look at some drugs in different classes. For example, the abused drugs, classic drugs of abuse. Pretty much almost all of them impact the dopamine system. And again, here's our dopamine system here, this pathway that you see right here. Right, here's the nucleus accumbens, which is a site very rich in dopamine, right? Medial forebrain bundle, right? And the axons that go from the nucleus accumbens to the cortex of the brain. You get sustained bursts of dopamine in the nucleus accumbens, right, in response to abused drugs. And a lot of this is occurring as a consequence of the inhibition of the neurotransmitter GABA in the brain. Okay? So almost all abused drugs stimulate dopamine release in this area of the brain, the nucleus accumbens. And indeed, that's been a focal point for studying the impact of various drugs of abuse on the addiction process, something that we'll get into in one of our later lectures. Stimulant drugs, crack, efficacy, cocaine, Ritalin, Adderall. These drugs, again, are categorized based upon their primary action, which is to cause excitement, elevated levels of alertness, motor activity, elevation of mood, and they all pretty much are impacting the dopamine receptors in the brain, what we call the type 2, type 3, and type 4 receptors within the brain. Actions of some stimulant drugs, amphetamines, again, they're stimulating dopamine synapses. It's causing more release of dopamine from that presynaptic part of the neuron. Cocaine blocks the reuptake of dopamine, which means that more cocaine is available to act on the postsynaptic area. Ritalin, which is used for the treatment of attention deficit disorder, that blocks reuptake of dopamine, but it does so in a more gradual kind of way. And again, this is a drug that very frequently is used for individuals that are suffering from attention deficit disorder. Actions of some drugs obviously can be very different depending upon the dose, whether it's a high dose or a low dose. Take the drug efficacy, for example, that increases the release of dopamine, but really only at low doses. And that really is responsible for its stimulant effects, its stimulant properties. Efficacy increases the release of serotonin at higher doses, and that is principally responsible for the hallucinogenic component of this particular drug. So again, efficacy, depending upon the dosage, can have very different effects. And that's something that certainly we need to be cognizant of with respect to any drug. Specialized effects of the drug nicotine. You know, many of us recognize nicotine as being the primary component, the primary ingredient of tobacco. Nicotine stimulates very widely in the body in what we call acetylcholine receptors. We call these actually nicotinic receptors. So when you take a look at nicotine and you take a look at what happens when it enters a physiological system, it can have effects on many, many different parts of our body, including the brain, obviously, because there are many of these receptors, nicotinic receptors, which are primarily receptors for acetylcholine. They are stimulating those nicotine is stimulating those receptors. So again, they're found throughout the central nervous system. They're found in skeletal muscles. There are many, many that are in the nucleus accumbens, which is very interesting because, of course, that's a site, part of the brain that's rich in dopamine. Nicotinic receptors are very abundant, okay, in that nucleus accumbens, and one of the things that they do is they start to promote dopamine release, which of course is involved in the experience of pleasure. So again, very interesting work in terms of these nicotinic receptors within the brain. Here's a little sidelight. Here's a real recent advance in this area in terms of addiction, the addiction that people develop to smoking. And of course, in the United States, it's been a huge problem for a long time. But 25% of the US population uses nicotine. And 6 million smokers worldwide die as a consequence of smoking every single year. A treatment, nicotine patches, gum, they're simply not effective at all in terms of the treatment. So there's been a number of researchers now that have been working with what we call a nicotine vaccine. And that vaccine absolutely could be game-changing. And here's principally how it works. A vaccination involves injection of what's called a hapten, and that hapten binds with the nicotine molecule. So when that occurs, then that molecule is very large, and indeed that molecule now can be perceived in our bloodstream, and the antibodies that make this nicotine now too large to cross the blood-brain barrier. So indeed, this is a very important advance that maybe young children will be given a nicotine vaccination at some point as part of the standard vaccinations that they receive. So again, this is an interesting sidelight of all of this, and it's one that is futuristic. It's one that perhaps may be developed to a point where we have a vaccine that absolutely could be game-changing in the addiction field. Opiate drugs, again, derived from the opium poppy. Opiates decrease sensitivity to pain, and indeed here are some of the primary abused opiates: heroin, morphine, codeine, fentanyl, mepridine, oxycodone. I think we're all familiar with these as being some of the primary abused drugs. We have in our brain what are called the endogenous endorphins. They have the same biochemical structure, almost the same as opiates. Those endogenous endorphins or endogenous opiates may contribute to certain kinds of pleasure and reinforcement, euphoria, for example. And the way in which they work is principally to inhibit the release of GABA. It's very interesting that GABA, inhibiting GABA means that you're going to promote the release of dopamine. So those endorphins are attaching to the very same receptors that opiates attach to if you consume opiates. Things like runner's high, for example, that we've known about for a long time. A person that's involved in a challenging kind of physical event like running, for example, you start to produce these painkillers, these endorphins. And if you push yourself and you're pushing yourself physically to new limits while you're running, then you start producing more and more and more of these endorphins. And pretty soon what happens is you get hooked on that and you push yourself even further in order to produce more of those endorphins in order to experience their pleasurable effects. So again, this is something that is called runner's high, and it is due to these endogenous opiates. We call them endorphins. That are produced when you're in a physically challenging kind of a situation, like running, for example, distance running. There's also what we call cannabinoid receptors. The psychoactive component of marijuana is something that's called THC, tetrahydrocannabinol. That's the active ingredient, the psychoactive ingredient of marijuana. Well, here's what this THC does. It attaches to very specialized receptors in the brain that are called cannabinoid receptors. And we see them in many, many different parts of the brain, in the cerebral cortex, in the cerebellum, basal ganglia, hippocampus, a lot of different sites within the brain. So that's what THC does, and that indeed is how you experience those pleasurable kinds of effects. So take a look at the location of cannabinoid receptors in the brain. Again, huge distribution here: brainstem, cerebellum, spinal cord, hypothalamus, amygdala, nucleus accumbens, basal ganglia, the neocortex. Again, many different areas of the brain. And those cannabinoid receptors, if they're activated, can produce a lot of different subjective effects, like loss of time, intensification of sensory experiences, memory impairment. Again, cannabinoid receptors, they're located, throughout the central nervous system, and they're involved in inhibiting the release of glutamate, inhibiting the release of GABA, and hence, increasing, dopamine release in the brain. So that's why you're getting those subjective kinds of effects that occur. Hallucinogenic drugs, they cause distortions in terms of perception. Here's some common ones: certainly LSD, certain types of mushrooms, angel dust, peyote. They resemble serotonin in terms of their molecular shape. So they're stimulating serotonin receptors within the brain. And indeed, this is causing, obviously, subjective changes in terms of what a person is experiencing. If you take a look at some very interesting incidents of, consuming various natural plants, like mushrooms, for example, they contain, some of them contain a substance which is called psilocin. And here's the molecular structure of psilocin. Take a look at the serotonin molecular structure, and it's very similar, right? Psilocin and serotonin are very similar to one another. So in certain parts of the world, and one part in particular in Mexico and other Central American countries, some South American countries as well, these mushrooms are harvested, and they're used for medicinal purposes. They're used to purposely distort a person's perception, maybe to have a higher level of religious experience, for example. But they act like the neurotransmitter serotonin. So they're stimulating serotonin receptors in the brain, and indeed they're producing this distortion in terms of perception. Here's another interesting example of modifying a person's experience, subjective experience, by way of modifying your neurotransmitter system. They're in certain countries, namely in parts of Asia, certainly in Japan, individuals will isolate themselves on these religious pilgrimages that they take, where, for example, in Japan, there's a sacred area, a sacred mountain area, where they climb to the top of this mountain, and they isolate themselves for an extended period of time, and oftentimes they experience hallucinations and distorted perceptions. And one of the things that we know is that when that isolation, associated with that isolation, is a very fundamental change in terms of serotonin. If you take a look at red blood cells, you see enhanced level of these serotonin receptors on these red red blood cells, and that's an indication. This is taken from an individual that has just recently gone through this sensory deprivation, and again, this is what is happening in those red blood cells, and presumably what is happening in your brain is that these serotonin receptors are dramatically increasing. So research then by scientists has suggested that this serotonin change may be very important for that, for those distortions in perception and hallucinations that those people experience. Here's another interesting part of all this interesting example, and that is what happens in terms of LSD. LSD binds on serotonin receptors in the brain. Here's some artwork that has been done by some Haitian artists that were using LSD in order to do their artwork, what we would call visionary artists, and they use this as a tool to try to get to another level of their consciousness, to experience things that they wouldn't ordinarily experience. And LSD, what we know about it in terms of its biochemical effect, it binds with serotonin receptors, and it overloads the brain. This musician that you see here, Paul McCartney, considered to be the greatest singer-songwriter in the history of music, readily admits to having used LSD when he wrote the lyrics for a number of the songs that he created. So you're allowed really to go mad for a little while and then come back again. It's like a telescope into the mind, and this is what LSD can do. I would not want you to think that I would advocate it at all. I don't, but this is what some musicians and artists, for example, use in order to create work that they wouldn't ordinarily be able to create. Lastly, let's talk a little bit about this researcher right here. His name is Emil Carcaro. And he's been working with patients that suffer from having a short fuse and are constantly getting into trouble. One such situation is that of what's called road rage, where a person will just completely fly off the handle and act very emotionally and aggressive towards another individual. One of the things that we know that Prozac does is to elevate levels of serotonin in the brain. And there's a lot of research that's been done by many different scientists that show that low levels of serotonin are associated with high levels of aggression. So in prison populations, for example, those individuals that are most aggressive tend to have lower levels of serotonin than those individuals who are not exhibiting high levels of emotion and aggression. So when Carcaro treats these individuals with Prozac, he's able to lower their aggression, and it has a very beneficial impact upon them in terms of lowering their overall level of emotion. Indeed, those individuals tend not to have those angry outbursts and emotional outbursts later on after being treated with Prozac. So Prozac, one of the things that it does, and we'll learn more about this when we get into the area of mood disorders, especially depression, is it increases levels of serotonin in the brain. It's almost considered to be a wonder drug in that it has, many different effects upon a variety of different behaviors. But again, Carcaro has had great success in treating certain types of patients with this particular drug. So that brings us to the end of this lecture, and what we're going to do in our next lecture is we're going to begin to talk about some important standard kind of methods that are used in the field of behavioral neuroscience, so that you have some familiarity with them as we go along.